Part of the reason you don't want to do notice. Row 4 has 4 seats. Seriously. That's spotted that from the top. Anywhere in
But not in those, just in this one.
okay. Thank you.
like it in the aisle. Just right aisle library.
Sorry, thank you.
Okay, good evening, everyone. Welcome. Please take a seat. My name is Matthew Scott. I'm president of the Carnegie Institution for Science, and I'm very happy to see you here despite all the beautiful weather out there. We're going to have more fun in here even than they are. To a paleontologist, fossils are a means to study links between living and once living organisms. To a geologist, fossils are a way to organize Earth's history. It was fossils, in fact, that enabled geologists to begin building their geologic timescale, an effort that has been in progress for hundreds of years. The Danish scientist Nicholas Steno provided the foundation in the mid-1600s when he recognized that sedimentary rock layers and the fossils they contain represent slices of Earth's history. by all means, come in and sit down. Don't hesitate. We want you here. 100 years later, after Steno, William Smith, a British surveyor, a real pioneer, found that by correlating fossils in one location with those in another, he could identify rock layers of similar age. Some of you may know the book, The Map That Changed the World, by Simon Winchester, which is about Smith. It's a very, very interesting story and a very well done book. So from this early work and since, paleontology and geology have been forever linked. And that's a nice reflection of some of the range of Carnegie science, I have to say. Now, when Charles Darwin proposed his theory of evolution, which he finally published in 1859, paleontology was still a young science. The word had not even been coined. But the fossils of extinct organisms that Darwin found on his voyage to South America on the Beagle, especially the giant sloths and armadillos, inspired him. They made him see relationships between living and once living species, and they provided some of the most direct evidence for his theory. They also presented the biggest hurdle. Darwin noted that Earth's rock layers must be full of fossils that show transitions between species. Had to be. But initially, geology and the fossil record did not reveal them. The lack of any, quote, finely graduated organic chain, unquote, as he wrote in The Origin of Species in Chapter 9, is perhaps, he said, the most obvious and gravest objection which can be urged against my theory. Just 2 years later, in a timely discovery, the fossilized bones of Archaeopteryx were discovered in Germany. This fossil exhibited traits common to both an ancestral group, the dinosaurs, and its descendants, birds. It was a missing link found and helped cheer him up quite a bit. Since the discovery of Archaeopteryx, hundreds, maybe thousands of transitional fossils have been unearthed. Neil's going to get up here and say tens of thousands probably, including notably a whole series of whale ancestors that illustrate the steps back to the sea. Among the most intriguing and important transitional animals is one called Tiktaalik. The first fossil Tiktaalik was discovered just 10 years ago in a remote part of the Canadian Arctic by our guest tonight, Dr. Neil Shubin, and his colleagues. Tiktaalik is a fish tetrapod bridge organism, sometimes nicknamed the fishapod. That's not related to the iPod. It illuminates the evolutionary transition from water to land. Tiktaalik soon appeared on the cover of Nature magazine, the important science journal, and in newspapers around the world, the front page of the New York Times, and it has since made its way into major textbooks and into our imagination of worlds past. The discovery of Tiktaalik in a rocky embankment far from coffee houses might appear to have been a stroke of incredible good luck. But it wasn't a particularly surprising or serendipitous discovery, at least not for Dr. Shubin. He knew something like it existed. Evolution and the morphologies of slightly older and slightly younger fossils, more like fish or more like amphibians, predicted it. Because of the intimate relationship between geology and paleontology, he knew which rock layers to examine. Geological maps showed him where those layers would be accessible on the surface, so off to northern Canada, in a 375 million year virtual journey. It took multiple arduous expeditions and many, many mosquitoes, and then there it was, a jaw protruding from a ledge. Dr. Shubin is a paleontologist, evolutionary biologist, and anatomist who has spent his career studying fossils, particularly those of fish and amphibians, and learning how they relate to the deep history of the human body. He has conducted fieldwork all over the world, and his work has been published widely. He is off to the Antarctic this year in search of fossils from a different era than Tiktaalik. Currently, he is the Robert R. Bensley Professor of Organismal Biology and Anatomy at the University of Chicago. He also serves there as the Associate Dean of Organismal Biology and Anatomy and is Professor on the Committee of Evolutionary Biology. He received his undergraduate degree from Columbia University and his master's and PhD degrees from Harvard, which is right up the stream from MIT, which is a much more interesting place in some ways. I have a few MIT connections. We got to take our chances when we have them, right? So, after a postdoctoral position at Berkeley, he spent 11 years, and I'm from Stanford too. This is really getting bad, so I'll stop. Okay, after a postdoctoral position at Berkeley, he spent 11 years from 1989 till 2000 as a professor of biology at the University of Pennsylvania. For 4 of those years, he was also a research associate at the American Museum of Natural History in New York City, and in 2001, he moved to the University of Chicago. At the same time, he became a research associate in the Department of Geology at Chicago's Field Museum of Natural History, where he served as provost from 2006 to 2008. A member of the National Academy of Sciences and a fellow of the American Academy of Arts and Sciences, Dr. Shubin is also a celebrated science writer. He has received numerous awards for the book he wrote in 2008 called Your Inner Fish, A Journey into the 3.5 billion year history of the human body. This book examines how much of your anatomy makes sense only in light of you having had fish ancestors. Nobody's upset. Good. Okay, great. The awards include the 2008 Phi Beta Kappa Book Award in Science and the 2009 National Academy of Sciences Book Award. The book was selected as the best science book of the year by Amazon.com and earned for Dr. Shubin the National Center for Science Education's, quote, Friend of Darwin, unquote, award in 2015. A 3-part PBS series about the book won an Emmy, as well as a 2014 Kavli Science Journalism Award and a 2015 National Academy's Communication Award. In 2013, Dr. Shubin followed up Your Inner Fish with another book, The Universe Within, The Deep History of the Human Body. When Dr. Shubin won the Darwin Award, no, not that kind of Darwin Award, the executive director of the National Center for Science Education wrote the following: lots of scientists do amazing science. Not many devote so much energy to making that science relevant to the outside world, and even fewer take on an area of science that is as deeply misunderstood by, and even frightening to, a very great number of Americans as is evolution. Now, of course, it should not be frightening, but wondrous and beautiful, and it is when it's conveyed by the right person. We salute Neil Shubin for being this rare scientist. Please join me in giving him a warm welcome.
Wonderful to be here. Thank you so much for that introduction. Matt, thank you for the invitation to be here. Thank all of you for coming out to hear about our inner fish. I should talk a little bit about how this whole concept was born. What's inner fish mean and where did it come from? It actually began, as you heard from Matt, I moved to Chicago in 2001. And I came as chairman of the anatomy department in the medical school. And my chief job as chairman of the anatomy department was to teach human anatomy to the first year medical students. And those of you who are MDs, or those of you who aren't, either know or imagine that this course is hugely stressful. It can be for many students. I mean, this is the first time they're learning about the human body and its structure in exquisite detail, memorizing thousands, if not tens of thousands of new names, doing this all over a cadaver, confronting their early career, confronting mortality. It doesn't get better than that, right? So to cool things out, what I used to do is hang around the tables with the students to let them get to know me and give me a chance to get to know them as we were doing the dissections, actually over the cadavers. So they almost invariably ask in those first few weeks, the students would come up to me, they'd say, Dr. Shubin, what kind of doctor are you? Are you a cardiologist? Are you a neurosurgeon? I'd say, no, I'm a fish paleontologist. And they'd be like, I don't know about. But it soon became clear that being a paleontologist, and not just any paleontologist, a fish paleontologist, is a powerful way to teach human anatomy. And the reason for that is oftentimes some of the best roadmaps to our own bodies lie in other creatures. Some of the best roadmaps to understand the structure of our brains lie in reptiles. Some of the best roadmaps to understand the cranial nerves, the complex tangle of nerves in our heads, lie in sharks and fish. And the reason for this, the reason why fish are such powerful tools to understand our bodies, is because we share a deep connection with them. We have a history. In every cell, in every gene, in every tissue, in every organ of our body lies artifacts of over 3 billion years of evolution. And that's our job to uncover, and that's what I want to talk about with you today. But the birth of your inner fish actually began, for me in a way, a lot before that. It began in graduate school. I remember I was a second-year graduate student, and I was fishing around for a new PhD dissertation. And I didn't know, I was really stressed, like, what am I going to work on? I had no idea how I was going to do this PhD thing. And the big step to do a PhD thesis is to actually find something to work on. And I was in class, and the professor showed this slide, this exact slide, from 1987, so you get a sense of how old I am. It is literally the slide that gave birth to several decades of my own research. What you have here is a cartoon of what we knew at the time about the transition from fish on top, that's a lobe-finned fish, a cartoon of a creature known from rocks about 390 million years old, that's when they first appear in the fossil record, to a tetrapod, an early limbed animal on the bottom. These are critters that first appear in the fossil record about 375 million years ago. I remember looking at this slide and thinking, holy cow, this is a first-class scientific problem. How did fish evolve to walk? I mean, when you look at the endpoints, it seems so utterly improbable, indeed impossible. I mean, fish live in water, right? And limbed animals and tetrapods live on land. How could this ever happen? So much has to change for this to take place. And so this really captured my imagination and my interest and my time, and it's been occupying my time ever since. I don't know what that says about me, that I've been working on the same thing for 30 years. But I began very simply as a paleontologist. So I approached this as a paleontologist would do it. And here's what you do. Look at this, and as a paleontologist, I want to find something like a flat-headed fish with fins, or fins with arm bones inside. I want to find a mix, an intermediate creature. So how do you do it? Let me give you the roadmap. There really, as Matt implied, there are 3 things you look at if you want to find a transitional form in the fossil record. You look for places in the world that have 3 properties. The first thing is you look for places in the world that have rocks of the right age to answer whatever question interests you. You know, remember I told you we're looking at the fish tetrapod transition. You got 390 on top is when they first appear in the fossil record, 365 on the bottom. So you want to be in a time period of the late Devonian period, within that 390 to 365 million year window. You know, if I was looking at the origin of mammals, I'd do something in the Triassic and Jurassic and so forth. So you choose the rocks of the right age to answer whatever question is of interest. The next thing you do is you look for rocks of the right type that are likely to hold those fossils. Not every kind of rock, as you can imagine, preserves fossils. Some are metamorphic and superheated and super squeezed. Others are volcanic and formed under the wrong conditions. Still others may reflect environments that the creatures never lived in or wouldn't preserve them as fossils. Okay, so as a sedimentologist, as a geologist, I wear that hat to try to understand what are the likely kinds of rocks to hold fossils. Finally, the third criterion, it does me no good if my wonderful rocks of the right age and the right type are buried 5 miles underground, right? They have to be exposed at the surface. We paleontologists love exposed, naked bedrock. Why? Because it's where the rock is exposed that we can see it, but also we can find the fossils at the surface. So when you open the pages of National Geographic, where are the paleontologists typically working? Deserts. We love deserts because that's bedrock central. Rocks of the right type, rocks of the right age, rocks exposed at the surface. Now you have the toolkit to find a major fossil discovery. It is very simple in concept. In execution, it's very difficult. There's a fourth criterion, and in my case, it was lack of money. So I started. It's so true. So I started my first academic job as Matt told you, at the University of Pennsylvania. So I'm here in the southeastern corner of the state of Pennsylvania. And as a young assistant professor, I couldn't really lead an exotic expedition. I didn't have the credibility to raise the money or to get the permits. I needed something local that I could do on turnpike tolls and gas money. There's something that I could do over weekends that I didn't have to do like long grant proposals for that would get rejected. You know, so I needed something local. So I dug out a geological map of the state of Pennsylvania, and I'm showing it to you here. I've stripped it of everything unimportant. And what you see is that Pennsylvania, 3 hours from my home here in southeastern Pennsylvania, is all this beautiful Devonian rock mapped by the Pennsylvania State Geological Survey. So really, I had lots of Devonian around there. I also got very lucky, and I began collaborations, friendships, that in science are so incredibly important. And I just wanted to show this slide now so I don't forget. Pretty much everything I'm telling you about on the paleontology side has been born of the people who have influenced me and I've had the privilege to collaborate with. This is true teamwork. I didn't discover Tiktaalik by myself. This was a team that did it with matching expertise. The person who showed me that slide, the late Farish Jenkins from Harvard, was my graduate advisor. He's been a partner in all this for many years. You could see we're wet, we're tired, we're in the Arctic, yet we're holding this beautiful thing here. There's a reason why there's a big smile on my face. And then with my ex-student, Ted Deschler, who was also a co-leader of these as well, Ted has been a partner in all this all throughout. Doing fossil work means having great field crews. We've worked over many years with fabulous field crews and wonderful lab people. So really, I want to underscore, this is true teamwork, okay? We work in the Arctic as a team, helping each other with complementary expertise. So in the, getting back to Pennsylvania, we had rocks of the right age, but the question is, do we have rocks of the right type? And it turns out they were perfect. If you want to think about what Pennsylvania looked like in the late Devonian, get Pittsburgh, get Harrisburg, get Philadelphia out of your brain, and think Amazon Delta. This is a cartoon of what Pennsylvania looked like 365 million years ago. You had highlands to the east, kind of where Scranton and Wilkes-Barre are today. You had an inland sea, the so-called Catskill Sea, to the west, more or less where Pittsburgh and Cleveland are today. That is, you have marine shales. And draining from east to west, the geological signature is of a series of rivers and streams that drain from east to west. Now, if you're a paleontologist interested in finding fossils at the cusp of the transition from life in water to life on land, this is perfect. Why? Because you can sample ancient seas. If you have the exposures, you can sample ancient estuaries, ancient rivers and streams. This is ideal. You have a range of environmental conditions and geological conditions that are highly likely to hold these critters. The problem is Pennsylvania is not a desert, okay? So the exposures that we worked on were not beautiful exposures. What they were areas where the Pennsylvania Department of Transportation decided to build new roads. What happens is, when PennDOT wanted to build a new road, what they would do is they'd come in with a crew and they'd blow stuff up, right, to make a road cut. And if I was really lucky, they'd make a road cut in Devonian rock, which is what they did here, and they expanded a road in the early 90s. This is called Red Hill because it's a hill and it's red. It's State Route 120. It's about an hour north of State College, Pennsylvania. There's a human being for scale right there. What you see are the geological layers or strata. If I was to show you a cross-section of these layers, these are the Devonian red beds. If I was to show you a cross-section of these layers, they'd look like a cross-section of an ancient river with, the lenticular in shape, with coarse-grained sediments like boulders and cobbles in the middle, fine-grained sediments on the border. You really see, in cross-section, you would see the life history of an ancient delta. Anyway, as soon as we got to these layers, this is about 3 hours from Philly. As soon as we got to these layers, kind of where this individual is, we started to find fossils. The first things we started to do were find fossils the size of railroad spikes. I mean, these were coming out of those rocks right there. We started to find jaws the length of your arm. These creatures, these first ones we were finding, were large carnivorous fish about 16 feet long, okay, with jaws the length of your arm, teeth the size of your thumb or railroad spikes. We started to find skeletons, like squashed ones like this, of lobe-finned fish. These are fish with lungs and gills that are closely related to limbed animals, to tetrapods. This is the side wall of a body. There's a squashed head. Then one year when I was up working on a dinosaur expedition, Ted visited the site and he discovered limb bones. He discovered limb bones of some early limbed animals. Like this is an upper arm bone, a humerus, of a creature identical to one that was discovered in Greenland in the 1920s, the late Devonian form. We've since discovered head bones, shoulders, leg bones, toe bones, got them all. We worked with National Geographic in the late 90s to do a reconstruction of that site in Pennsylvania, and this is kind of what that road cut in Pennsylvania looked like 365 million years ago. Think about it. Today, there's a road with trucks whizzing by, but inside the rocks is this world, which is this wonderful disconnect aesthetically. I always think that when I'm looking in different places. We had that large freshwater fish, that 16-foot long one. We had some of the earliest forest-like plants with small shrubs. We had a series of small armored forms, and then a series of species of limbed animals, tetrapods. So this is a Devonian world we were pulling out of this landscape in Pennsylvania. But Ted and I realized we had a problem, a big one. We were in rocks that were too young. Our Catskill rocks were 365 million years old. And in this portion of the late Devonian, we're already finding very derived limb bones, very derived tetrapods. If we look at the evolutionary trees that our colleagues were doing, if we look at the stratigraphic, the layers of the geological column, where other fossils were known, it was very clear we were about 10 to 15 million years too young. We had to push this whole thing back, rocks of the right age, rocks of the right type, and so forth, back about another 10 to 15 million years to a time period called the Fronian of the late Devonian. It'll mean something to you in a second. So to give you a sense, the whole expedition here was to try, what it began with, is to try to find a flat-headed fish with fins and fins with arm bones, to find a mix here. And we were, just to give you a sense of what we're looking for, just look at this, you get some key differences. One big difference is if you look at the lobe-fin fish on top, you see its head is more or less conical with the eyes on either side. If you look at an early limbed animal, let me go back. If you look at an early limbed animal, what you have is a flat head with eyes on top. You have similar bones that make them up, so they're clearly very similar animals, but the proportions and the geometry and the function is very different. So big differences in head. The other thing is you see a big difference is in the neck. That is, these fish don't have necks. They have a head that's connected to the shoulder by a series of plates of bone. Whereas if you look at an early limbed animal, what they have is a head that can swivel independently of the body. The head is not connected by bone, plates of bone to the shoulder, and there are specialized joints that enable the head to swivel. Another big, I could talk about these differences all night, but I just want to focus on these three because they're going to mean a bunch in a second, is, fish have fins with fin rays and with bones in the middle, whereas tetrapods, limbed animals, have arms and legs with fingers and toes and wrists and ankles. Ted and I were finding a lot of fingers and toes and wrists and ankles. We weren't finding flat-headed fish with fins or fins with arm bones on the side. So really to move back in time meant moving to the Fronian stage of the late Devonian, about 15 million years earlier. Okay, we pulled out the paleontological toolbook again, playbook again. Looking for rocks of the right age, rocks of the right type, rocks that are exposed to the surface. We had an idea to work in Brazil. We had an idea to work in the western part of Alaska. Everything changed serendipitously in my office one day in 1998 while I was still at the University of Pennsylvania. Ted and I were having an argument, a geological argument, about nothing related to paleontology. In fact, I even forget what the topic was. But to settle the debate, I pulled out my college geology textbook. This is it, Evolution of the Earth by Dott and Batten, second edition. I think it has new authors now. It's in its 17th edition or something like that. But anyway, we settled the debate. And I remember we're chewing the fat, and I'm just turning through the pages. And I hit a diagram that was to change my life. Diagrams changed my life, if you haven't noticed. But I hit one that really defined 13 years of field expeditions in a college geology textbook, and this is it. I want to run through it because it shows exactly what we look for. It says Upper Devonian sedimentary facies, and these are the rocks that remember that are in our right window of time. These are the sedimentary rocks that can be formed under the right conditions. What you see here is a map of North America. There's the United States in the center, there's Mexico down south, at the bottom, there's Canada up top. And superimposed on this map of North America is an estimate and a hypothesis of the environments that the Devonian rocks originally formed in. That is, the so-called depositional environments of these early rocks. So if you look at the western part of North America, it was an ancient seaway in the Devonian. There's all marine Devonian rocks out here. But then these authors showed three areas that were formed in ancient delta systems, much like the Amazon Delta, just like the Catskill rocks that Ted and I had such luck with. I think you see where I'm going. I'm showing them in red. The first one was these Catskill rocks. These are the red beds that Ted and I had worked on for several years. Been there, done that. We knew about that, so it sort of confirmed what I knew. There's another one here in eastern Greenland. Been there, done that too. I hadn't been there and done that. That was done by Swedes and Danes in the 20s and 30s. Do you remember that cartoon of the limbed animal I've been showing you a bunch? That was a cartoon of a fossil that was discovered by those teams working in Devonian rocks of East Greenland. I think you see where we're going. Extending 1,500 kilometers east to west across the entire Canadian Arctic was a series of mapped Devonian age rocks of the right age, of the right type, and if I had any luck at all, they'd be exposed because the Arctic is itself, in this place, a very large desert. We were very excited. I looked at Ted. I said, Ted, did you know anybody who worked on these rocks? He says, no, do you? I said, I just asked you that question. And we went back and forth. We were like, nobody works on these rocks. All right, so we ran to the library. This is the day when you actually had to go to the library in 1998. So we ran to the library, and we saw things in paper. It was amazing. And we discovered papers by this gentleman here, Ashton Embry. Ashton Embry, back in the late 60s and early 70s, was a mapping geologist. His job was to map the rocks in the Canadian Arctic, and he did it for years. What they would do is they'd drop Ashton off in the middle of the Arctic with an Inuit helper, with a sledge, with some dogs, with cans of food, and hopefully some can openers, and then they'd leave, and Ashton would then use his Brunton compass and do it old style mapping, which was really impressive. And as the context of this work, what Ashton did is he produced a paper which was so fundamental to our work, it really defined how and why we should work in the Arctic. And this is the paper I'm going to show you. The title is not going to be a bestseller. It's the Middle Upper Devonian Clastic Wedge of the Franklinian Geosyncline. This is the most important paper of my life, but for a lot of reasons. And the reason why I'm showing it to you is I get asked all the time, how do you know where to look as a paleontologist? And I say, oh, just go to this page of Ashton Embry's paper. It's right there. I say, what? Okay, when Ashton described the age of the rocks of this area, so he named these rocks the Fram Formation, Norwegian for forward, after a very famous exploration vessel that worked up there for a period of time. He said the age of the rocks of the Fram Formation are the available data indicating an age of what? Early to middle Franian. Remember the exact age that an understanding of evolution told us to work? Franian, bam, that's it. The S is silent. Then when he talked about what the Fram Formation rocks were like, this is what really got my heart going. The Fram Formation is similar to the Catskill Formation of Pennsylvania. Okay? I mean, it gets a little more complicated than that in terms of the stratigraphy and the sedimentology of it, but in a nutshell, that is exactly what led us up there and why we spent a lot of time failing before we were successful. Then he showed a picture of some of the rocks. This is the kind of exposure. This is the type section of the Fram. Very beautiful spot in terms of what to work on. This all happened in one morning in 1998. We're like, wow, I'm hungry. Yeah. So we ran to the Chinese food restaurant down the street from my office at Penn, and I had a fortune cookie, and I kid you not, I moved with me from Philadelphia to Chicago. It's on my door. It said, soon you'll be sitting on top of the door. The problem is I've never played these numbers. I probably should do that. Anyway, so now that we had a new set of problems, instead of, like getting in my Subaru wagon and driving, to central Pennsylvania, now we're working here. It's a little different working in the Arctic. It's daylight 24 hours a day. In the summer, it's dark 24 hours a day. In the winter, we have to bring all our food in. You're highly dependent on aircraft, as I'll show you in a second. There are polar bears up there, and that's a continual threat because polar bears will hunt humans, and that's not something I really like thinking about a whole lot. To give you a sense of how remote this is, the nearest village Inuit village to our sites is 300 miles away. It's a village of 175 Inuit. This is a picture of the big city of the Inuit village, Grise Fiord, Canada, in spring. Okay, so that gives you a sense of the logistic chain, the supply chain we really need. So much of the science we do is dependent on aircraft. And so we use these fixed-wing aircraft called Twin Otter. If you've ever been in the bush, you've been in a Twin Otter, I'm sure, because they have a very short takeoff and landing distance. We're beyond the tank of gas of a helicopter, so the twins will come in, bring in fuel, and then the helicopter will hopscotch to us. The reason why this is important, the reason why I'm showing this slide is these things are very weight dependent, and fossils are very, very heavy. So when we're up there for about 4 to 6 weeks, and it becomes a giant salvage operation, right? And we have to decide what comes and what stays. It's very difficult. We have to make these decisions on the fly in the field. So it's been very challenging, particularly in bad weather. To give you a sense, this is camp before we set it up. We don't bring a lot of stuff. So we don't bring a ton of people. We usually bring a kid from that Inuit village. They're really amazing to work with. All our food gets packed in these sealed tubs. We seal them up tight because the food can smell. So we don't want the bears, who have great noses, we don't want them to stay on the ice, not where our tubs are. So we've sort of honed the Arctic camping experience for us to avoid polar bears, but also to put us in sites that maximize our success. This is a colorized version of a diagram from Ashton's paper, Ashton Embry's paper. So on the upper left here, we're in Nunavut territory. There's the flag of Nunavut in Canada. There's the North Pole where my pointer is. This is Nunavut territory. If we zoom into Nunavut, right here, there's Ellesmere Island. I'm going to look on the right here, there's where Ellesmere is. See, it's this big island where my pointer is now on the right. And you can see, surrounded by red, these are where the Devonian exposures are. Okay, so there's a ton of Devonian. There's 100 kilometers for scale. This is a playground for us. To give you a sense stratigraphically, we're right here. We're in the Devonian, the Franian stage of the right Devonian. that is central casting of where we want to be. Okay, so we had the fortune cookie in 1998. It took us about a year to raise money and get permits. This is where we started in 1999. It was our very first set of field camps here in the western part of the Arctic. This is what camp looked like at the time. Each of us have our own personal tent. When you build a wind wall around these, they can withstand 70-mile-an-hour winds, which is real sweet. Unfortunately, the kitchen tent cannot withstand 70-mile-an-hour winds, so I've spent the better part of my professional career chasing this thing around the in storms. But this gives you a sense of how we work. We don't have to bring water in because we can drink water right out of the ice fields or the glaciers. But these are the Devonian rocks in the western part of the Arctic. They're very flat-lying, posed serious challenges for weathering. But essentially the research here is we quadrant out areas to work, and we literally crawl or walk over spots looking for places where fossils will weather out on the surface. So we're looking for signs of fossils on the surface, then we dig in. We had a big problem this first year in 1999. It was a complete flop. We lost 2 weeks to really bad weather, so we were stuck in these tents for 2 weeks. That was no fun. But also, even more importantly, we were in the wrong kinds of rock. We were finding deep water fish, fish like sharks and others that are known to be deep water fish in the Devonian. These were marine rocks from this middle of the ocean. These are not a place where you'd expect to find an early limbed animal. We were clearly in the wrong part, geologically the wrong part of the Arctic. So what did we do? We revamped. I'm going to show you, this is the slide from Pennsylvania, but it actually applies to the Arctic as well. We were in the middle of the sea, way out there. What we needed to do was go upstream, okay? Geologically, to go upstream, that meant moving east, okay? So the next year we went east to get into these more estuarian and river environments. And this is what it looked like in 2000. We're now in southern Ellesmere Island. It's quite a bit more montane, which is great for preserving rocks and fossils. You could scramble up and down these hills. It's not a whole lot of fun, but you can do it. And this is what camp looked like, new kitchen tent, which still found its way to get blown into the fjord. But we've actually solved the kitchen tent problem, but it's been the story of our lives to have bad kitchen tents. Anyway, as soon as we got to this spot, we started to find lobe-fin fish fossils. These rocks reflect ancient rivers and streams, much like Pennsylvania. And when we're in these rocks, we started to see cousins of what we're looking for, lobe-fin fish. Nothing I'd be here talking to you about. They were known elsewhere, but we showed we were on the right path. What we then had to do was, okay, we're in the right general environment, but now we needed really fine-grained rocks. We needed, not coarse-grained, we needed rocks that were formed on very fine-grained sediments, largely because it's those fine-grained sediments that can preserve beautiful fossils. And we had specific hypotheses about the kind of fine-grained rocks we wanted. That meant moving to another valley, and so it took another couple years to do that. I'm making 6 years look really fast. So we ended up in this valley here, and this is a valley where essentially an undergraduate, a college student who was working with us, discovered a bunch of fossil fragments on the ground. And it turns out that that layer, which he found fossil fragments on, was produced by hundreds of fish skeletons piled one on top of the other. So then our task became really quarrying this layer out, removing these skeletons as best we can to try to make sense of what's going on. Okay? So cut to the chase, back 6 years later, we worked this layer, and what we typically do is we'd work in a line. Each of us have a little spot, just like archaeologists, and then dig it out. My colleague here, Steve, in the blue, was working the layer. I was to the right here. We removed rock from right there, this spot right here. This is it. He removed rock and he said, hey guys, what's this? As soon as I saw that, I knew we had found what we had spent 6 years looking for. What you have here is everything red here is Devonian rock. See this V? There's a V right there. You see rock here and there's a little V, there's a little crack in it. I looked at this, and I'm just going to mean something to you in a second. I know you're looking at it thinking, well, this guy's crazy. But there's teeth here. It was an upside-down snout, and not just any snout. It was a snout of a fish, and I could tell that it was a fish because of the texture on this bone. And it was a fish snout upside down, but it wasn't just any snout. It was a snout of a fish, and it was a snout of a flat-headed fish. And you're going to wonder how I knew this. I'll show it to you in a second. But a flat-headed fish was sticking out of me. Remember I told you conical-headed, flathead? Here I had a flathead staring out at me. I'm like, wow. So we jumped up and down a little bit, and then over time, we removed the rocks around here. Steve removed a lot of it. You can see how the pedestals out, and then capped it with plaster and brought that whole bunch of rock home with this tip of the snout preserved. That was removed later. This gives you a sense of what it looks like from another angle. This is the dip of the rock. I mean, it's really tilted. This poor person here, oh, I feel so sorry for him, because he has Farish's boot in his face, and all the rocks are coming down. And the challenge is even more severe. This is where Steve was pulling out that specimen I was going to show you in a second. But there was another one of these flatheads down here, and there was another one of these flatheads that came out in here. So we started to find flatheads all the way down the road. To give you a sense of how painful it is, just look at his poor face right there. I mean, he's like, I so don't want to be at the bottom of the hole. But anyway, so these are the specimens here. They're wrapped in plaster. We now use urethane, and they get capped because they come back on the bottom of a helicopter, and the bottom of a sling for the long trip back to base, and then to be shipped home. That's a first-year University of Chicago graduate student for scale. Anyway, so you bring these things home, and a lot of the work here is to remove the rock initially. This is before we had high-powered CT scanners. I'll show you that in a second. But initially, the cast would be open, and then the rock would be removed grain by grain. So here is that specimen, Steve's specimen. This is in preparation. This is 4 months of work. This is the needle. He removed rock grain by grain. Look at it. Started to mow. It looks like a flathead. See, there's one orbit or eye hole. There's another orbit or eye hole. Looks like we're seeing a flathead. Another 8 months go by. Boom. Looks like you have a flathead with eyes on top. Look what's emerging here. There's one shoulder. There's another shoulder. Looks like we might even have a neck in this animal. Over time, this thing's is being exposed by a preparator. Remember, this whole expedition was to say 390 for fish, 365 for tetrapod. We want to find a flat-headed fish with fins. We want to find fins with limb bones inside. We look for rocks of the right age, rocks of the right type. We use an understanding of stratigraphy, of evolutionary biology, and this is Steve's fish. It has like a fish, it has scales in its back and fins with fin webbing. Like an early limbed animal, it has a flat head with eyes on top, has a neck, and when we crack open the fin bones, what do we see? Bones that correspond, as I'll show you in a second, to upper arm, forearm, even parts of a wrist. This is a cast, a high-fidelity cast of this specimen right here. Okay, so it gives you a sense of the scale of the thing. So this would be about 4 feet long. Now, to show you that V, I mean, it's the reason why I'm bringing this is so you can see how you train your eye to do this kind of thing. I'm sorry, I'm going backwards, but it's necessary evil. So here's the V, right? There's the V right there. See, there's the V. There's the V right there. So that's the skull. So what was sticking out of it is this V. There are the teeth there. And here was the flat head. And this is what was exposed over time. Now, while this was being exposed, there was a trial going on in Dover, Pennsylvania. Intelligent design creation, as it was trying to be taught in school. And people were saying there are no transitional fossils in the fossil record. And this was sitting on my desk. All right, let's go cut to the chase here. So just to give you one more anecdote, and then we'll get into the science of the thing. So as discoverers of a new species, you get to give it a name. That's one of the great privileges. And so what we decided is, since we were working up there at the privilege of at the courtesy of the Inuit people, we started a naming project with the Inuit elders, and this is the Inuit elders here that we worked with. And we had 2 requirements for the name. We really wanted to be engaged with the community. One was to have a name that's meaningful to them and to us. And the other was to have a name that we could pronounce. The name of the committee didn't lend me a whole lot of optimism. We had a name that we could pronounce. So I was talking to this gentleman. I was in Chicago. He was in Grease Fjord, actually. And it proved to be very difficult to have a name that was meaningful to both sides. I said, look, we found a fish. Okay. We found a fish in the rocks. Long pause. Hunters don't find fish in rocks. Oh, right, no concept for fossils. They didn't have a concept for fossils. So we had to work around that. It took some time, but eventually they had seen stuff like this in rocks before, so we were able to leverage that a bit. So the fossils was okay. Then they got worried that this thing was worth a ton of money. And I'm like, no. It's about science. It's about knowledge. It's not about money. Then he thought I was crazy. Okay, so we had all this go back and forth. Finally, he's like really very frustrated with me. He said, just tell me what it is and where it lives. I said, well, it's a large freshwater fish. He said, why don't you say so? You got yourself a Tiktaalik. I said, a Tiktaalik, what's that? He said, it's a large freshwater fish. Well, that was easy. So the Tiktaalik it was. And so in 2006, we had multiple specimens. We now have over 20 individuals of this thing. They range in size from about 4 feet long to about 9 feet long. And just to get you a sense about what we knew in 2006, we now have discovered much of the back of the specimen to the base of the tail from multiple specimens. Has a flat head with eyes on top, right like an early limbed animal. There are configurations of the bones themselves that are part fish, part limbed animal. I don't need to get into that, but it really has a mosaic of features there. This is a fish with a neck, clearly a neck. It's a very tetrapod, limbed animal-like neck. Massive shoulder girdle, huge ribs, clearly had both gills and both lungs. It has fin rays. You can see the fin rays here, a little bit reduced, but they're definitely there. I'll show them to you in the other slide. And the limb bones has one bone, two bones, has limb bones that compare to early limbed animals. Now, so much has changed since we discovered Tiktaalik in 2006. Technology has evolved to the point where we can now image rock, and we can use high-energy CT scanners. Not the kind you'd want to use in a doctor's office, but high-energy CT scanners to look inside the rocks and see what's inside. And this gives you a sense of one of the limb bones. You can look at, here's the humerus, there's the radius, there's the ulna. You can see individual limb bones in there, the fin rays. We can model where the vasculature is. We can model from the shape of the crests and the bones, the musculature of this thing. This is a fish that has multiple, so we can actually begin to take apart the joints. This is the shoulder joint of this fish. There's the socket on the shoulder. There's the ball on the humerus. This is a fish with an elbow. This is the radius and ulna, radius on the right, ulna on the left. You can see the shape of the articular surfaces. And then here's the distal or far away surfaces of the humerus, or the elbow of this animal, clearly set to do a form of bending and a form of pronation, that is rotation of the thumb side, as it did some flexion. And there's two wrist joints, which compare to early amphibians, which are what known as the proximal carpus and the distal carpus. These are two wrist joints that are involved in flexion. We also can scan the head. This thing was clearly a predator. So when we take this head here and we pop it in the CT scanner, we can really see the tusks and the teeth and so forth, and we can really see the sutures inside. So you can see that big teeth, large fangs. This is clearly a fish-eating fish. We now can model the biomechanics of how this skull worked, because we can see the sutures in here and make estimates of how they moved. So technology has really given us a whole new window on this thing. And we can begin to really think about this creature in its environment. That is, what you have in Tiktaalik is clearly a very aquatic creature, right? It's a creature which has gills. It has fins, but it's also capable of living on land, in the mud flats, maybe partially excursioning on land, because it has limb bones and able to support itself with its appendages. It has lungs to be able to breathe air. It has a biting style and a shape of a head that could function like a crocodile or an alligator or what have you. Tiktaalik gets most of its meaning, and this is where the inner fish piece comes in, most of its meaning not in isolation. It gets meaning when we start to compare it to the other fossils that are known from around the world. When we look at creatures, and Tiktaalik's in the center here, when we look at creatures with these complicated names like Eusthenopter and Panderichthys and Acanthostega and Ichthyostega, what we see is a gradual transition from definitively a fish to definitively a limbed animal with real fingers and toes. And we can look at the shape of the head and how it evolves. We can see how the shape of the fin evolves and so forth. It's part of a continuous series. Now, the story of Tiktaalik is really interesting. It tells us a lot about the transition from life in water to life on land. But one of the key take-home messages of the talk is that this transition, this great evolutionary event that happened over 375 million years ago, is not just some esoteric event in the history of life. This is an event that's embedded inside our own bodies. This wrist, for the first time, that we see in Tiktaalik and its evolutionary cousins, is something, when we look at the fossil record, is to become our own wrist. This neck we see for the first time in Tiktaalik and its evolutionary cousins is something that will become our own neck. That is, every time you bend your wrist and every time you shake your head and countless other features, you can thank creatures like Tiktaalik and its cousins evolving in Devonian streams. Seems strange, but how do we know that? Easy. We could trace the record. That is, I could trace the evolution of a humerus from a basal fish to Tiktaalik, to an amphibian, to reptiles, to mammals, to other primates, to people. I could do that with the radius and ulna. I could do that with the wrist bones. We can use the fossil record to show the continuity of forms. And then we can trace, using the fossil record, our arm bones to the fins of fish. We could trace the bones in our skull to gill bones in fish. But it's more than fossils. We can use the record of developmental biology, of embryology, going from an egg to an adult. We can use the record written in DNA of living creatures. And we can probe that to understand how we're connected to other animals, including fish and worms and so forth. It's a beautiful story because we can now begin to pull in multiple lines of evidence to understand our connection to the rest of life on the planet. It changes you. I'm a fish guy, right? So I'm a little messed up. So you look at this, and you see a pinnacle of human achievement, Albert Einstein. I look at this and see a version of a big fat old fish. You can compare Professor Einstein to the fish. I labeled it here. Einstein's on the left, by the way. You can compare Professor Einstein to the fish, and we can do it with the fossils to show the continuity, but we can do it in development. Let's just take a little riff on the development here. If you look at the human head a few weeks after conception, what you have is this, okay? You have a series of paired primordia for the eyes. These are where the eye and the lens and the retina will develop. Then you see a series, in this case, 4 swellings. They're paired swellings that develop in what will become the pharyngeal area. I've color-coded them for you. What you have in these swellings are cells. Some cells that are migrating into there, others that are dividing in place. But these cells are forming these clear-cut swellings, and there's a little cleft between each swelling, and there's other structures on the inside that are very important to understand our own heads. What happens in sharks and fish? Here's a shark. The embryo is not identical, but look, you have paired primordia for the eyes, and you have these paired swellings. You can even color-code them in similar ways. What we can do is we can actually map, we can label those cells, and we can ask the question, what do those cells become in fish and in people? Okay. Well, in fish, that first light blue one, like in the shark, become portions of the upper and lower jaw. The others, in dark blue through yellow, become portions of the large portions of the gill apparatus. And this includes the skeleton, the muscles, the nerves, and arteries. So really the whole thing is related to this initial segmental organization is reflected in the structure of the gills of a shark. What happens in people? That first one becomes a lower jaw and 2 bones in the middle ear. That second one, the dark blue one, becomes a little throat bone called the hyoid, and then 1 bone in the middle ear. The other 2 become throat bones and portions of the voice box, as well as the muscles and nerves and bones that control all this stuff. What does this mean? What this means is many of the muscles and nerves and bones I'm using to talk to you with right now, and many of the muscles and nerves and bones you're using to hear me with right now correspond to gill structures in sharks and fish. How do I know that? I know that by comparing the developmental fates of these cells. I know that by comparing the DNA that instructs these organs to develop, that instructs those cells what to become. I also know it by collecting fossils. That I can follow a fossil trail to show you how this dark blue bone here, called the hyomandibula, second arch gill bone in a shark, becomes the stapes of the human ear. Fossils, genes, comparative anatomy, embryology. It's a story that takes us to wonderful places, showing how much of our head is originally evolved in gill structures in sharks and fish. But it gets more than that. If you think about one of the great sort of problems of developmental biology, it's been the centerpiece of Carnegie science for quite a time, is understanding how this fertilized egg, a single cell, becomes a body that goes from a single cell to become each of us here, with 3 trillion cells or so of our own, 3 trillion cells, all packed in the right place, all differentiated. We call that process bodybuilding, to go from the thing on the left to the thing on the right. But this is the challenge of developmental biology. How does this happen? What instructs these cells to form this pattern, to differentiate into different tissues from a single cell? How does that happen? Locked in understanding that is really understanding a lot about evolution. Because here what you're dealing with in the egg is in the egg to go from this creature on the left to this creature on the right is really an interplay between the proteins that are inside the egg that are from the maternal, from the mother, but they're also based on the DNA inside of the individual that will develop from this egg. And it's that interplay, and that interplay of those, the proteins and the DNA, with the environment over time, where there's a DNA recipe of a common toolkit of genes oftentimes that gives rise to a body. So locked in this is also not understanding the basics of our body and how it forms, but understanding the differences in how a fish forms from an egg to adult, to understanding how a human forms from egg to adult, to understand what the differences are, both genetically and developmentally. Well, let's talk a little bit about that, because this is actually part of the work of my lab. I'm going to show some unpublished results, but this will give you a sense of what we work on. So here, if you look at, I'm going to finish with this toolkit piece. Here's a zebrafish on the right. It's a living fish, okay, related to trout and salmon and so forth, the so-called teleost. And here's a human on the left, and there's an arm. Now look at this. If you look at their skeletons, you don't really see an inner fish here, right? You only see the inner fish when you compare the fossils. And indeed, if you look at the skeleton of a fish fin, look at this. It's got these long gray bones. These are the fin rays. They're made of a certain type of bone. Then he has these black bones here at the base. They're formed out of cartilage. But if you look at the cartilaginous bones of a fish, and you compare them to the cartilaginous bones of a human arm, no similarities there. You could look. But here's what's amazing. When we start to look in the DNA that builds them, builds the fins, builds the limbs, we start to find a signature of the human hand inside the fish fin. Let me give you an example. One class of genes, which actually Matt Scott worked on and made some seminal discoveries in, are the so-called Hox genes. And these genes are actively involved in defining regions of the body. So here's one. Here's a mouse on the right. You see a mouse arm on the right. And here's one of these genes, the so-called Hox gene. And it shows what you see here is where the gene is active in the limb bud of a mouse. Now it turns out if you look at this activity, this little paddle here is what will become the wrist and digit. So if you map these cells and you trace what these cells are going to become in the adult mouse, look at this. These cells that contain this gene mark the wrists and the fingers almost precisely.